So the last point of sexual reproduction that we're going to be looking at in this area of study 1 is the biological advantage of sexual reproduction, specifically the genetic diversity in offspring. So when we talk about meiosis, and we had that version of crossing over, we also talked about the importance of the centromeres still being joined at metaphase 1. And when meiosis goes wrong, occasionally a pair of chromosomes fails to disjoin or separate at the anaphase correctly, so that either 2 copies of a chromosome rather than a usual 1 are present in a gamete, or a copy of the chromosome is missing. So we have that disjoint from the centromere. This event is termed a non-disjunction, and it's an unpredictable error. Non-disjunction can occur at either step 4 or 6 of meiosis, and we're going to go through that in a minute. So this is what non-disjunction looks like. So you can see that we have some homologous chromosomes. We can see that we have a couple that look similar. Up here, these two look quite similar. They're the same length. These two again look quite similar. These two, and we have homologous chromosomes up until we have our X and our Y. So these are our sex chromosomes. So they're not part of what we're talking about here when meiosis goes wrong, but that's just a visual example of why that's called a Y, because you can see the Y there. And then we've got our normal X-type chromosome. Where we have gone wrong is this pink one down the bottom here. We have a third chromosome here, and this is typical of Down syndrome. So someone who has Down syndrome will have an extra chromosome. The condition is caused by trisomy, which is so 3-somy at 21, which is the presence of an additional copy of the chromosome 21. So our 21st chromosome hasn't split apart properly. So this is what it can look like. Normal meiosis is here on the left-hand side. So we're looking at meiosis 1. We've still got those 2 there. We end with a 2N. We go through mitosis 2, and we end up with these individual 4 times N. And we have 1 lot in each. Remember, because we're trying to create 4 gametes. We have a non-disjunction at meiosis 1. So this means that they're both pulled, say, to the same side of one cell. Yeah, so we have all of our copies only in this side and not in this side. So this is a non-disjunction at meiosis 1. And this results in 2 of the 4 gametes having 2 sets of chromosome 21. So this is how it can happen. Whereas the other side here, it doesn't get any of that. It doesn't have that chromosome at all. So these two will be non-functional. When we have a non-disjunction at meiosis 2, we're looking at meiosis 1 has gone past well. It's all sorted, well and good. But meiosis 2 is what we're having trouble with. Meiosis 2, their anaphase hasn't split them properly because we have perhaps an issue with the joining at the centromere, and we result with 2 normal ones, but then 1 without and 1 with. So this is a really good case study that you could have in your repertoire for when we come to exam period. So have a look at these in a little bit more depth. Make sure you understand what the consequences of meiosis 1 versus meiosis 2 non-disjunctions look like. And what are the options? So you might be given this here at the bottom, and you might have to look at, hey, what's gone wrong here? So think about how you can do that. So non-disjunction should be one that you add to your cue cards. A deletion removes a chromosomal segment, duplication repeats a segment, an inversion reverses a segment, and a translocation moves a segment. So these are some other issues with meiosis. So a deletion, you can see here, has removed D entirely when it's gone to replicate. A duplication means we've got a double up. So we've got BC here, but when we get to a duplication, this half is twice as long now, or has duplicated B and C again. Inversion is when we flip these loci or loci along the gene. So you can see here they're no longer in order. And translocation moves a segment from one chromosome to another, but a non-homologous one. So when we're getting that crossing over happening, it's not happening correctly. And this is a reciprocal translocation. So that means they've had to swap each into one another. So while we have these issues and we have the breakage of the chromosome and we have the chromosomal parts that are affected, this also creates genetic variation. And genetic variation is something that's quite important when we talk about sexual reproduction. So while it might look like these are flaws, they're actually not always. These give rise to genetic variation, and they can change the way that we are programmed. So they can change our phenotype, which is our observed genotype, which we're going to go through a little bit later in the unit. But these, while they are issues in meiosis, are really important for genetic diversity, which is what we're talking about in this last dot point. We also want to have a look at polyploidy. So remember, ploidy was one of the words that we had up on the whiteboard the other day to talk about adding to your cue cards. Having more than 2 sets of a chromosome in a genome is called polyploidy. And this is as a result of an error in meiosis and can affect the ploidy in the zygote. In humans, polyploidy zygotes don't survive, but polyploidy can result so it can be a result of mitosis as well. So think about it in regards to your M checkpoint. So when we're at that metaphase checkpoint in our cell cycle in mitosis, we're thinking about, are they lined up correctly? Is everything attached properly? If you think about, if only one of our centromeres is attached and we go through anaphase, it's going to pull back. And we're going to have that polyploidy occurring in one side of our 2 cells that we're about to produce. Now, polyploidy is common in plants because many plants can survive using asexual reproduction. So triploid plants are usually sterile or have low fertility. Some banana plants have been found to be triploid. So cotton and potatoes are also tetraploid, which is 5. And then bread wheats are hexaploidy, and strawberries are even octoploid. So you need to understand ploidy and what that means. And it means as another set of chromosomes. So we have that trisomy when we were talking about Down syndrome, whereas this is ploidy. So this is 3 sets or 4 sets or 5 sets or 6 sets or 8 sets in the whole genome, not just the one. So polyploidy can result in more vigorous plants and being able to bred into horticultural populations. And so we can think about this as an advantage over vegetative propagation. So remember, that's where we just split off and we start growing a new other half of the plant. So when we're looking at that 3rd dot point, we need to have a look at how meiosis can go wrong and how we can give rise to genetic variation through that. But we also need to look at the disadvantages. So some of our advantages are we have a long-term evolutionary potential. We have the ability to adapt, I guess, reasonably quickly based on external conditions. Because we're so genetically diverse, some of the population is going to survive. There is unfavorable genetic variation, and that's eliminated from the population more efficiently. So that's a really good thing. So when we talked about back here in this slide, when we have polyploidy, we said that this doesn't really survive, especially in humans. It also generates genetic variation, selects for beneficial genetic variation more efficiently. So things that are better for us, our ability to breathe perhaps, or our lack of allergies, or say if we have issues creating our blood from our stem cells, that kind of gets bred out of the population pretty quickly because people carrying those genes are unlikely to reproduce as efficiently as those who carry healthier genes. And this means that populations are better able to adapt and survive in changing environmental conditions. And this is a massive driver of evolution. The disadvantages, however, is that we have a slow reproductive rate. We have fewer offspring produced over a longer time span. So if you think about the way that we reproduce, we undergo sexual reproduction. So we need to wait to find a mate. We need to have a mate that's suitable. We need to have a long gestation period. We need to have proper gametes that are produced all the time, that are healthy, that we can then take advantage of when we do find that mate. Recombination can also break apart beneficial genomic combinations and introduce deleterious variation to populations. So this is when we were talking about these kinds of things, when we had a duplication or an inversion or maybe a translocation, and we're looking at how that can vary populations, and sometimes that can be pretty bad. The potential for spread of sexually transmitted diseases is also through a population. So we have sexually transmitted diseases they are also looking to just reproduce their genome. So if they can pass on into a new host, that's beneficial for them, obviously not beneficial for us. So that's the spread of disease can become quite quick. And they talked about that a lot with the Tasmanian devils. They thought that their interactions were spreading the cancers on their face. Sexual reproduction is also energetically costly, so it requires a lot of ongoing energy input from the parent. So not only does that mean creating our gametes, so women who are born with all of their, or they have all of their eggs at the start of puberty, and they lose them slowly. If you think about how many times we lose those, which is once a month, that's wasted energy that our body has created. And males are kind of lucky. They only recreate sperm when they're, running on empty. So they are actually creating a lot of energy, and perhaps not always for the goal of creating an offspring. So that's something else that we need to look at, is that it is costly to create that every single time just in case. So that's a lot of energetically costly stuff for us. We might not notice it, but if you think about other animals, that's something that they actually factor into as well. So now that we check off all of our dot points, you should now know how offspring from two parents has a unique genetic identity. So that's through our crossing over and recombination, remembering those are two separate processes. The key events in meiosis that result in the production of gametes from somatic cells, including the significance of crossing over of chromatids between homologous chromosomes in prophase 1, so that's where we get that unique genetic identity, and the non-dividing of the centromere in metaphase 1. So that's when we're talking about our trisomy. We need to make sure that we're still connected and we're still going through to split into our 4 haploid cells by the end of both of our meiotic divisions.